Hello everyone and welcome back to C.A.R. Entertainment. I'm your host Dylan Hamilton and we are going to be talking about the biochemistry of photosynthesis today, guys, in our continuation of A22 biology revision videos. So far we've talked about the actual process or actual parts of photosynthesis and the external factors which affect the rate of it, but this is the main video you're going to have to watch, is the biochemistry of photosynthesis. So please, if you're watching this, please try to link it and share it to people, because this is very important. It's actually more important than the photosynthesis video, because that just gives you an introduction to the absorption spectrum and the action spectrum. But this is actually about the actual individual parts of how photosynthesis takes place through light harvesting, the light dependent reaction, and the light independent reaction. We'll go through it right now. So, like I said, there's three different stages. Light harvesting stage, which is the absorption of light in the thylakoids. The light dependent stage, which is when energized electrons make ATP and NADPH, and this takes place in the thylakoid membranes of the grana. You then have the light independent stage, which is the products of the light reaction or the light dependent reaction are used to make carbohydrates, and carbon dioxide will be fixed as a part of the cyclical reaction of series of reactions that take place in the stroma. So let's go through light harvesting then. Well, chlorophyll, as we know, is arranged in clusters, and each cluster is called an antenna complex. Chlorophyll is the primary pigment of this antenna complex, and it is located at the base of the complex called the reaction center. The antenna complex will harvest light over a range of wavelengths, as we know, due to the presence of different pigments such as chlorophyll A, chlorophyll B, and carotene. The light energy absorbed throughout the complex will then be passed down by special proteins. This absorbed energy will pass down by special proteins to the adjacent molecules, and this is known as resonance transfer. So there's molecules all in the complex, and the energy is passed down to adjacent to all these molecules through special proteins. So like we said, the energy is funneled in the direction of the reaction center through resonance transfer, but this resonance transfer requires molecules in the antenna complex, and these have to be part of a regularly arranged structure. So like we said, that molecules and how they're fitted down. So each antenna complex you find forms a photosystem, and a photosystem is very important because there's two photosystems for every light dependent reaction, which we'll now go through. So the light dependent reaction, well, electrons will be excited in this thing called photoactivation. That's quite important. There's two photosystems that take place in the light dependent. Usually for every cell you have your two photosystems. So you have photosystem 1 and photosystem 2. We're just going to call them PS1 and PS2. They will differ in proportions of different accessory pigments that will make up the cluster in the antenna complex. So photosystem 1 will have chlorophyll A, and will have an absorption peak of 700 nanometers. So we're going to call PS1 with PS or P700. But PS2 also contains chlorophyll A, but its absorption peak is 680 nanometers, maybe because it absorbs less red light, for example. And we're going to use it as P680. So the excitation of electrons will result in the electron transfer pathway described as a Z scheme. So let's go through the process and the light dependent stage then. Well, light will firstly be trapped in photosystem 2. Now bear in mind, when I go through this and I talk about this, this has happened simultaneously. Photosystem 1 and photosystem 2 are happening simultaneously. They are happening at the exact same time. So if you get an essay to write about photosynthesis, including light harvesting, light dependent, and the light independent stage, whenever you come to the light dependent, just say, as a wee note, photosystem 1 and photosystem 2 are taking place at the exact same time, and then you can just write out this method. Because it's happening at the exact same time, but for the purpose of me teaching this, I am going to talk about a linear format because it's easier for you to understand. But just know that it happens at the exact same time. If you do not know that this happens at the exact same time, you'll be getting 0 marks for this. So just make sure you know that it happens at the exact same time, the process of photosystem 1 and photosystem 2. So light energy is trapped in photosystem 2, and it will boost electrons to a higher energy level. These electrons are then received by an electron carrier, and the electrons are passed from an acceptor. This electron carrier is an acceptor, and it will be passed along a series of cytochromes. The electron transfer is coupled to the synthesis of ATP and phosphorylation, as we know from in respiration. So then the electrons that are passed along the acceptor along the series of cytochromes, which are coupled to the synthesis of ATP and phosphorylations, these electrons replace those lost in photosystem 1, because photosystem 1 also loses electrons. So they gain their electrons back through photosystem 1. Okay? The electrons lost by photosystem 2, because obviously they've lost their electrons that eventually go around to replace photosystem 1's, their lost electrons are removed from water, and the resultant photolysis of which produces O2 and hydrogen plus ions. So their electrons that they lose are removed from water. They eventually come in and form water, but they're removed in photolysis, and that produces oxygen and hydrogen plus ions. Okay, and that's essentially then how photosystem 1 gets them back. So light energy is trapped by photosystem 1, and it will boost its electrons to an even higher level than in photosystem 2. Bear in mind it's taking place at the exact same time. The electrons are then removed by an electron acceptor, like in photosystem 2. But this time the hydrogen ions from the water that we know combine with the electrons from the second electron acceptor, and these reduce NADP to NADPH. So essentially, in photosystem 2, their electrons that are lost make ATP in phosphorylation, and they also make water, but it is then going through photolysis, which produces O2 and hydrogen plus ions, and these hydrogen plus ions combine with water, with electrons in water, to form NADPH, as it's summarizing from NADP. So at the end of the light dependent stage, you have ATP and NADPH. Then in the light independent stage, which occurs in the stroma, it will use the NADPH and the ATP to fix carbon dioxide to carbohydrates, which is what the plant's planning to do after all. Photosynthesis is to create energy, and that's what it needs, carbohydrates. So NADPH provides a reducing power, and ATP the energy source. So the light independent reactions will incorporate the cyclical metabolic pathway called the Kelvin cycle. And this is the fixation of CO2, because CO2 will enter the stroma and combine with a 5-carbon compound ribulose bisphosphate, RuBP, just known as RuBP, and that'll be fine. So when CO2 enters the stroma and combines with RuBP, then it will form 2 glycerate phosphates, otherwise known as GP. But this reaction is catalyzed by the enzyme Rubisco, or ribulose bisphosphate carboxylase, just known as Rubisco. So glycerate phosphate is then reduced by ATP and NADPH as ATP and NADPH converts GP into triose phosphate, or TP. And we know this because it also happens in respiration. So ATP and NADH will reduce it into triose phosphate from glycerate phosphate. But ATP and NADP that is formed will also leave the stroma and then enter the granule lamellae whenever they've reduced the glycerate phosphate. RuBP will then be regenerated because 5 out of every 6 triose phosphate molecules that are formed will regenerate BP, and this will also use the remainder of the ATP that are there. So if you produce 15 triose phosphates, you will get 3 RuBPs, because you just divide it. So if you have 15 triose phosphates and it's 5 of every 6 that are produced, then you'll know, or 18 would be the better format there actually. So if I said you have 18 triose phosphates, you want to get 3, because obviously 5 of every 6. So if I asked you how many molecules of TP would need to be produced to synthesize 5 RuBPs, you just do a quick calculation of 5 out of 6 using your fraction. So the product synthesis then, TP will be generated to produce starch, glycerol, fatty acids, and amino acids, with nitrogen being obtained from the absorbed nitrate ions as well. So just know that. So what is the actual link between the light dependent and light independent stage? Well, because light is removed, the light dependent stage will stop. Okay, that can't carry on without light because it's dependent on it. And therefore ATP and NADPH will not be made. And glycerate phosphate will not be converted to triose phosphate, and therefore there won't be a buildup of GP. There'll be a reduction in RuBP as a result, because RuBP fixes CO2 to form GP. Okay, so what that actually means is, because light is removed and the light dependent stage stops, that means ATP and NADPH is not made. Therefore, the light independent stage can't actually happen. So although it doesn't need light to occur, it still can't happen, because it doesn't have any ATP or NADPH to convert the glycerate phosphate to triose phosphate. It can't do that without the ATP and NADPH made in a light dependent stage. So with CO2 no longer available and not being fixed, and GP not being formed, RuBP will build up and GP will be converted to TP, and it will rapidly fall. So that's essentially the link. I've been Dylan Hamilton from C.A.R Entertainment, and I'll see you next time.